Welcome to Chapter 9, Section 6, Media Used for Bacterial Growth. Learning Objectives. Well, okay, it's just one. Learning Objective. Identify and describe culture media for the growth of bacteria, including examples of all-purpose media, enriched, selective, differential, defined, and enrichment media. The study of microorganisms is greatly facilitated if we are able to culture them, that is, to keep reproducing populations alive under laboratory conditions. Culturing many microorganisms is challenging because of highly specific nutritional and environmental requirements and the diversity of these requirements among different species. Nutritional requirements. The number of available media to grow bacteria is considerable. Some media are considered general all-purpose media and support growth of a large variety of organisms. A prime example of an all-purpose medium is tryptic soy broth, TSB. Specialized media are used in the identification of bacteria and supplemented with dyes, pH indicators, or antibiotics. One type, enriched media, contains growth factors, vitamins, and other essential nutrients to promote the growth of fastidious organisms that cannot make certain nutrients and require them to be added to the medium. When the complete chemical composition of a medium is known, it is called a chemically defined medium. For example, in EZ medium, all individual chemical components are identified and the exact amounts of each is known. In complex media, which contains extracts and digests of yeast, meats, or plants, the precise chemical composition of the medium is not known. Amounts of individual components are undetermined and variable. Nutrient broth, tryptic soy broth, and brain-heart infusion are all examples of complex media. Media that inhibit the growth of unwanted microorganisms and support the growth of organisms of interest by supplying nutrients and reducing competition are called selective media. An example of a selective medium is microch9part6 agar. Oh wait, that's pictured right here, which I've used before, but I don't use here. It contains bile salts and crystal violet, which interfere with the growth of many gram-positive bacteria and favor the growth of gram-negative bacteria, particularly the Enterobacteriaceae family. These species are commonly named enterics, reside in the intestine, and are adapted to the presence of bile salts. The enrichment cultures foster the preferential growth of a desired microorganism that represents a fraction of the organisms present in an inoculum. For example, if we want to isolate bacteria that break down crude oil, hydrocarbonoclastic bacteria, subsequential subculturing in a medium that supplies carbon only in the form of crude oil will enrich the culture with oil-eating bacteria. The differential media make it easy to distinguish colonies of different bacteria by a change in the color of the colonies or the color of the medium. Color changes are the result of end products created by interaction of bacterial enzymes with differential substrates in the medium, or in the case of hemolytic reactions, the lysis of red blood cells in the medium. In this figure, the differential fermentation of lactose can be observed on microch9part6 agar. The lactose fermenters produce acid, which turns the medium and the colonies of strong fermenters hot pink. The medium is supplanted with the pH indicator neutral red, which turns to hot pink at low pH. Selective and differential media can be combined and play an important role in the identification of bacteria by biochemical methods. A medium that is selective against gram-positive and differential for lactose fermenters. If it's a lactose superfermenter like Escherichia coli, it will acquire this green metallic sheen that you can see pictured here. Otherwise, if it ferments lactose, at least some, you'll get this this lighter purplish color there. Another example of a differential and selective media is mannitol salt agar. So it is selective against bacteria that can't tolerate a higher salt concentration, and is selective. And it is selective for organisms that are able to metabolize mannitol, the mannitol salt. You create a product that changes the pH, as in B here. We also have this combination tube option, a triple sugar iron. I'm not going to go through all the explanations of this right now. And these are carbohydrate tubes where you can differentiate acid production, acid and gas production, or alcohol fermentation based on whether the inverted small tube, the Durham tube on the inside, has a gas bubble in it or not, and whether or not the media is pH is changed so that the pH indicator turns yellow. Case in point, the end-of-year picnic. The microbiology department is celebrating the end of the school year in May by holding its traditional picnic on the green. The speeches drag on for a couple of hours, but finally all the faculty and students can dig into the food. Chicken salad, tomatoes, onion salad, and custard pie. By evening, the whole department, except for 2 vegetarian students who did not eat the chicken salad, is stricken with nausea, vomiting, retching, and abdominal cramping. Several individuals complain of diarrhea. One patient shows signs of shock, low blood pressure. Blood and stool samples are collected from patients, and an analysis of all food served at the meal is conducted. Bacteria can cause gastroenteritis, inflammation of the stomach and intestinal tract, either by colonizing and replicating in the host, which is considered an infection, or by secreting toxins, which is considered intoxication. Signs and symptoms of infections are typically delayed, whereas intoxication manifests within hours, as happened after the picnic. Blood samples from the patients showed no signs of bacterial infection, which further suggests that this was a case of intoxication. Since intoxication is due to secreted toxins, bacteria are not usually detected in blood or stool samples. Well, not the pathogens, I guess, aren't detected in the stool samples. You're going to still detect a crap ton of bacteria in there, pun not actually intended. microch9 agar and sorbitol microch9 agar plates and xylose lysine deoxycholate XLD plates were inoculated with stool samples and did not reveal any unusually colored colonies, and no black colonies or white colonies were observed on XLD. All lactose fermenters on microch9 agar also ferment sorbitol. These results ruled out common agents of foodborne illness, Escherichia coli, Salmonella species, and Shigella species. Analysis of the chicken salad revealed an abnormal number of gram-positive cocci arranged in clusters, as pictured here. A culture of the gram-positive cocci releases bubbles when mixed with hydrogen peroxide. The culture turned mannitol salt agar yellow after a 24-hour incubation. All the tests point to Staphylococcus aureus as the organism that secreted the toxin. Samples from the salad showed the presence of the gram-positive cocci bacteria in clusters. The colonies were positive for catalase. The bacteria grew on mannitol salt agar, fermenting mannitol, as shown by the change to yellow in the medium. The pH indicator in mannitol salt agar is phenol red, which I'm sure you're all familiar with from chemistry labs, which turns to yellow when the medium is acidified by the products of fermentation. The toxin secreted by Staphylococcus aureus is known to cause severe gastroenteritis. The organism was probably introduced into the salad during preparation by the food handler and multiplied while the salad was kept in the warm ambient temperature during the speeches. What are some other factors that might have contributed to the rapid growth of Staphylococcus aureus in the chicken salad? Why would Staphylococcus aureus not be inhibited by the presence of salt in the chicken salad? All right, that brings us to the end of Chapter 9. See you again for Chapter 10 or whatever I do next.